Thank you very much for having me here today. I would like to talk about bridging bold and electrophysiological signals. One of the important questions is to understand how bold signals measured with fMRI and electrophysiological signals measured with EEG, MEG, or intracranial EEG can inform about typical and atypical processing in the human brain, such that we can learn about how the human brain processes information. But one of the issues with these type of measurements that we have to consider, take into account when we are doing these measurements, is that these human brain measurements are at the population or mesoscale. And that under 1 millimeter, there are already 100,000 neurons. So the question we have to ask is, how do these human brain population measurements integrate information across large populations of neurons? And the way in which I would like to discuss that today is by looking at fMRI and electrocardiography signals and see how they respond differently to visual input, and to think about what type of visual information these signals encode. And then I would like to talk about which type of neuronal activity they emphasize and what the differences are between these measurements. And then how that is important to identify potential biomarkers for neurological diseases, such as photosensitive epilepsy. So the first part, I would like to talk about field potentials. Field potentials can be analyzed in many different ways. And there's many interesting parts of the field potentials, and I would like to discuss two of them today. First, I would like to talk about gamma oscillations, which are oscillations in the field potential with a repetitive timescale between 30 and 80 hertz. And when we look at the power spectrum from such an oscillation, and the frequency is on the x-axis and the log power is on the y-axis, you can see this peak in the power spectrum because there's a particular time scale. And what this indicates about the underlying physiology is that there is something synchronous in the underlying physiology, that neurons are receiving inputs or firing at the same time. And these are thought to be generated by circuits of excitatory and inhibitory neurons. And I would like to contrast that with broadband signals that were considered later in the early 2000s. And broadband signals are, for example, when there's a field potential, it has a random walk shape, which in the power spectrum, if we look at the frequency and the power, has a 1/f distribution. And these broadband signals are modeled and have been correlated with Poisson input firing rates. So when the neurons are talking to each other at very different timescales, spanning the entire power spectrum. And to think about these two different concepts, gamma oscillations or like asynchronous broadband processes, I would like to do a little experiment. And what I would normally do if it weren't a COVID-19 period, I would ask you to think about the synchronous gamma oscillations when we are doing something synchronous in a large group of people. So I would ask you to, on the count of 3, to tell your neighbor about the current month of the year. So 1, 2, 3, July is what we would all say. And then I would contrast that with doing something random, such as tell your neighbor the month of the year in which you were born. And go 1, 2, 3, and everybody would tell their neighbor something different. And when we're listening to everybody, we can't really hear what they're saying, but there's a lot of information exchanged. So when something asynchronous is happening, you can still see a large signal increase because a lot of information is conveyed, but we will not be able to pick up on the population scale what kind of information that was. So I would like to see the synchronous all doing the same thing at the same time with the gamma oscillation, but asynchronous signals can increase in a broadband manner where there's a lot of different things going on. And one of the questions that we had to ask early on in our research with ECOG is about gamma oscillations, because gamma oscillations and their synchrony have been proposed to play important roles in vision. For example, carrying feed-forward information and being essential for conscious perception. But we almost never saw these gamma oscillations when we measured human ECOG recordings in motor cortex or in higher order areas in the ventral temporal lobe. We only saw these broadband signals. So a question that we wanted to ask is, can we actually measure these gamma oscillations with human ECOG? Or is this something special that we don't get at the population scale? And we look back at the literature, and what we saw is that many studies that measured gamma oscillation in visual cortex used stripes or grading patterns, because Jubal and Wiesel showed that oriented edges activate neurons in visual cortex strongly. So grading patches drive visual cortex very strongly. And we'd never used a grading. So we had a patient that had electrodes implanted, ECOG electrodes implanted for epilepsy monitoring for clinical purposes. And there were some electrodes on early visual areas. The seizure didn't come from there, but there was a clinical question about those areas, so they placed electrodes there. And we went in and we asked these patients, we have IRB approval to ask these patients to participate in research. And we asked the patient to look at images on the screen and showed this patient to grading. And this is the a single trial from an early visual recording, and you see this nice evoked potential early on in the signal. And then you see these clear gamma oscillations in a single trial in ECOG electrode. And also in the power spectrum average across trials for this electrode, you can see this very clear narrowband peak around 50 hertz that is increased, particularly for the stimulus compared to the baseline, which is plotted in gray dashed lines. Here's frequency, here's power. So you can see it's around 60 hertz. So we found gamma oscillation with ECOG. It's not due to the measurements. It's just the fact that we had to show gradings. But when we showed other images, such as noise patterns that also have contrast and are known to increase firing rates in early visual cortex, we only found, you can still see the ERP, but we only found these random walk responses and clear broadband increases spanning the many frequencies. So images drive the ECOG signals in different ways. But when we look at these gamma frequency bands, this one range can have many different shapes. In one case, it is a peak. In the other case, it's flat. So we have to distinguish these different components to be able to understand what they tell us about the images. And we wanted to understand what types of other features were reflected in these images. Like, what about other image features? Is this just gradings, or what's going on? And so we showed this, the patients some objects. And you can see here again that we get two reliable components in the ECOG signal and that they differ across images. You see this left car on the left, and this is the power spectrum for baseline in gray and for this particular car in red. And you can see that there's a clear broadband increase for this car. And it was reliable across the trials. And this other car has a clear broadband increase, but also a narrowband gamma increase. So it's not only gradings. So the question is, there are different components in the field potential, and they have to be considered separately, but what do these broadband and narrowband gamma components encode in terms of visual processing? And in order to understand that, we showed the subjects 86 different images. You can see here from left to right. We used different complex squiggly line patterns that occupy different parts of the visual field from left to right, from bottom to top. Gradings of different orientation, gradings of increasing contrast, plats of increasing contrast, images with 8 orientations of increasing contrast, these complex squiggly line patterns of increasing contrast, images change in sparseness, incoherence, and noise. And what I'll show you first is the fMRI signal recorded from a representative voxel in early visual areas that was published by Kendrick Kay and collaborators. And they measured the percent signal change for every image. So every bar here reflects the fMRI percent signal change for one image. So you can see that this voxel had large responses to, didn't have large response to image on the left, but did have large responses to image on the right. Didn't have large response to image on the bottom, but to contrast patterns on the top of the field, but had larger responses to these complex patterns compared to gradings. And then also the fMRI signal increased with contrast, as you can see here, for gradings, plaids, 8 orientations, and these complex patterns. And we presented the same images to 3 subjects who had ECOG electrodes on early visual cortex, and in which the ECOG electrodes were not involved in epilepsy. And we extracted the broadband component of the ECOG signal. And you can see that this component matched the fMRI signal very well. You can see that in terms of the complex patterns had a larger signal compared to these grading patterns. And then the signal increased similarly for with increasing contrast. So we used the image computable model that was developed for fMRI, and we applied it to the ECOG broadband signal. And on average, across the electrodes, we could explain 80% of the variance cross-validated across images. And so ECOG broadband encodes very similar visual features as fMRI. And we would like to contrast that. I would like to contrast that when we extracted the narrowband gamma increase for every image. Because you can see that these patterns that would elicit an enormous broadband response barely elicited any narrowband gamma response, and most of these were within the noise. However, there was large gamma increases for the gradings and then increases for larger contrast. But it's very different. Narrowband gamma encodes different features. So we had to change the model that predicts the ECOG narrowband. And we changed it to encode variance across orientations, such that a single orientation is present and others are not. So there's a large variance across orientations. It drives the gamma signal strongly. And it's sensitive to contrast. So more contrast, larger variance, more signal. And we could explain 75% of the variance across the electrodes, across the 3 subjects in early visual cortex. So these different components encode different parts of the visual input and can tell you different things about how these images are processed. So different field potential components encode different stimulus features. And how can they differ so much? What do these components tell us actually about the underlying neuronal activity? How come these features differ so much? Why is the fMRI signal different from the narrowband gamma signal? Why does it match the broadband signal? Can we explain these relations? So I would like to show you a computational framework of how we think about currently about integrating these signals. And a typical approach that we have taken and many others have taken is to directly correlate signals. So we directly correlated ECOG with fMRI signals, and we have learned a lot from this approach. And we found that the fMRI bold signal often correlates well with ECOG broadband signal, as you also saw in the previous slides. But bold does not correlate with narrowband gamma. An additional variance in the bold signal was explained by low frequency power changes measured. And in order to take a different approach than just correlating these signals and saying like one matches, one doesn't, we want to think about how each signal integrates across a population. So we developed a oversimplified but hopefully useful computational framework. So let's first think about how these different signals integrate activity across a neuronal population. So let's imagine 2 neurons that have the same distance and same orientation compared to an ECOG electrode. And we think about the membrane potentials in each of these neurons. And then we could take the sum of the time series to approximate the field potential measured with this electrode. But if we would think about how much energy each of these membrane fluctuations would use, we would have to think about whether they go up or down or drift away from their resting potential. Each of those fluctuations would require energy. So we could, for example, think about the power as an approximation for the energy that is used. So let's, in this example, let's assume like this and this all is using one energy, and we can take the sum of the power to approximate the fMRI signal. And what you can see here is that if we would change the phase of one of these two neurons, then that would have a large effect on the sum of the time series or on the field potential signal, but it wouldn't affect how much energy is used by each membrane potential fluctuation. So synchrony has a large effect on ECOG and on other field potential signals, but not on fMRI. So how can we test this framework? Let's first consider these two equations. So ECOG and fMRI encode fundamentally different aspects of neuronal processing. And if we take ECOG to be similar to the power of the sum, if we will take the ECOG power spectrum, and fMRI to be similar to the sum of the power, then these two are related in a very nice way mathematically, because the power of the sum is equal to the sum of the power plus the sum of the cross power. And this shows that the ECOG signal is driven by both synchrony, which affects the cross power, and activity, which affects the sum of the power. While the fMRI signal is not sensitive to neuronal synchrony. And whenever the green term is large, these two may not match anymore. So whenever there's a lot of synchrony, it changes from stimulus to stimulus, from condition to condition, ECOG and fMRI signals are going to disperse. So how do we test that? So first we had an ECOG experiment where the visual stimuli changed broadband gamma and low frequency power changes. And we simulated membrane potentials that produces these three signals based on existing hypotheses, and we fit that to the ECOG data. So we simulated membrane potentials to produce broadband gamma and low frequency power changes and fit the amount of power change to how much it changed in the data by taking the sum of the time series of the membrane potentials. So we have a simulated set of underlying neuronal activity, and then we tested whether simply taking the sum of the power would predict the measured bolt signals. And the simulated bolt predicted the measured bolt signals and explained about 80% of variance in V1. And moreover, it showed that ECOG gamma and simulated gamma could be large, but it didn't predict bolt. And moreover, broadband correlated positively and low frequency power correlated negatively with bolt, both in the data and in the simulation. So field potentials are sensitive to synchrony and fMRI is not. And taking that synchrony into account in our models of what drives an ECOG and an fMRI signal is important to explain the similarities or the differences. And it's also important to consider these different aspects to understand how these different mesoscale measurements can inform about neurological diseases. And one of the things that when I talked about this research with some of my colleagues in neurology, and I showed them that these gratings induce gamma oscillations, they told me that these grating patterns can actually trigger seizures in photosensitive epilepsy if it was a large grating on the screen. Like what? Grading patterns that are used so often in vision research can actually trigger seizures. So we went back and I looked at a lot of the research about what type of visual features can induce seizures in photosensitive epilepsy. And we found that based on the literature review of the last several decades, that a lot of different visual features have been compared. And for example, when you increase a grading in size, the number of epileptiform responses in the EEG increases. However, if we think about the fMRI signal, it actually decreases with increasing size due to surround suppression. And when you change contrast, orientations, luminance versus red-green chromatic, increases spatial frequency from low to middle, middle to high, grading edges, or noise patterns, the epileptiform responses all change in a particular manner based on the literature. And fMRI signals do not match these responses. They change in a very different manner. For example, they decrease with size. They don't change much and increase maybe a little bit for a plat compared to a grading, and etc. Whereas gamma oscillations show a very similar pattern and respond in a very similar way to visual input. And firing rates, again, change in a different way compared to the epileptiform responses. So it's important to understand these different components. And what we hypothesize based on this, the fact that the same visual stimuli that drive gamma oscillations and epileptiform responses is that they are driven by the same circuit. And that the circuit that is involved in gamma oscillations may actually also be involved in photosensitive epilepsy. And what that that means that our knowledge about gamma oscillations is not just about a particular feature in early visual cortex, but it actually may help us understand diseases. And models developed to predict the amplitude of gamma oscillations may then also predict which stimuli are provocative. So with the help of the software gaming project at the University of Utrecht, we implemented the model that I showed you earlier of gamma oscillations in a smartphone app. So what you see here is the screen of a smartphone that is processing the camera input, and each square here gives the output from the model of gamma oscillation. So it predicts what the potential gamma power would be for an area of cortex that would respond to that part of the image. So you can see here that some of the image have a very low likelihood of gamma oscillations, but some parts of this input may be very high. And for example, that escalators that are known to be very provocative in photosensitive epilepsy and even cause headaches also in migraine are detected and are likely to induce gamma oscillations. And other patterns in our environment as well. So different field potentials components encode different stimulus properties. And field potentials are sensitive to neuronal synchrony, and fMRI is not. And understanding these mesoscale signals is fundamental to identify potential biomarkers of neurological diseases. The neuronal circuit that drives gamma oscillations may also be involved in photosensitive epilepsy. And for these projects and these multimodal integration, we think it's very important to share our data in a fair manner. And we have approval to share our anonymized data for most of our studies. And in order to facilitate the reuse of data by others, we built on the brain imaging data structure that was initially developed for MRI and then extended to MEG. And we worked closely with EEG BIDS extension to extend it to intracranial EEG data. And Chris Holdgraf and a lot of our collaborators worked really hard to make sure that it fit well with all the different aspects of intracranial data that we encounter. So we can now share our data in the same way as MRI studies are shared. And we can develop standardized computations to work with ECOG data. And if you want to learn more about any of the studies I showed you and you can actually get the data and code and reproduce the published figures with these papers. So all the code is available on GitHub. And there are links to the Open Sciences framework where you can download the data. And you can rerun some of the analyses. For example, the PLOS biology paper, you can rerun the simulation where we simulated the computational framework about the differences between bold and two potential recordings. Thank you very much for having me here, and thank you very much for your attention. I would like to thank my collaborators, Kai Miller, Brian Wandel, Kendra Kay, John Winnewer, and Natalia Petridou, Nick Ramsey, and Dorothy, and also my the clinical intracranial EEG teams that have helped tremendously in terms of collecting the data. They're always a big support to this type of research, and of course my funding. And thank you very much for having me here.
So hi everyone, and hope you're all doing well. First, I wanted to thank the OHBM organizers and everyone involved for their work in making this a virtual meeting. So this lecture is going to focus on basics of simultaneous EEG and fMRI and how combining these techniques can let us expand the range of questions that we can ask about the human brain. So we're going to discuss ways in that EEG and fMRI are complementary to each other and motivate reasons for why we want to combine them. Then go into some basics of the data acquisition, some of the main types of artifacts that can arise when you try to gather EEG in the scanner, which can sometimes be quite challenging to deal with. And then we'll go through some examples of EEG fMRI studies and at the same time walk through some different strategies for analyzing the data. So one motivation for combining these technologies is that they're very complementary, both in terms of their spatial and temporal resolution, as well as what physiological aspects of brain activity they're measuring. So a nice thing about EEG compared to fMRI is that it's a direct electrical measure. And in this talk, we'll be focusing on scalp EEG, which uses a cap or a net of electrodes sitting on the head to measure voltages on the scalp, rather than putting electrodes directly inside the brain. So even within just a second of data, oops, where's my mouse? As you can see, we can see oscillations that are happening much faster. So for example, this is called the alpha rhythm, which is an 8 to 12 hertz oscillation that you can usually see when someone closes their eyes when they're relaxed. However, the spatial resolution of EEG is relatively low because it's not straightforward to localize where in the brain are these sources coming from that are generating the voltage patterns that we see on the scalp. And also signals get more weak and dispersed before they reach the scalp. So individual neurons are much too weak to detect. And it turns out that our EEG signal is coming mainly from larger synchronized neural populations oriented perpendicular to the cortical surface. Now in bold fMRI, we're measuring changes in blood oxygenation that's coupled to local changes in neuronal metabolism. So it's not the electrical activity directly, but is an indirect response to neural activity. Oops, sorry, I keep losing the mouse. And this vascular hemodynamic signal is slower and more delayed than many types of electrical phenomena. So our intrinsic temporal resolution is lower. Though in the next talk by Laura Lewis, you'll hear more about how EEG fMRI can help us to also study faster fMRI responses. But fMRI has a number of advantages beyond EEG in that we can better resolve where the signals are coming from in space and across the whole brain. Our spatial resolution is on the order of millimeters and even below, and we can even resolve very fine structures like cortical layers and columns. So ideally, it would be great if we can combine the best of these two worlds and try to get better temporal resolution using EEG, along with the improved spatial resolution we have with fMRI data. And luckily, we have technology that lets us gather these at the same time. So we can put someone in the scanner while they're wearing the EEG cap and be able to record their scalp electrical signals and their bold fMRI data at the same time. But we also know that we can do EEG and fMRI separately, and that presents fewer technical challenges, as we'll see. But I would say that the main benefit of doing these at the same time is that the signals will correspond to exactly the same neural and experimental settings. So you're capturing the same brain states that the person's transitioning through. Everything about the environment is the same. The subject has the same amount of habituation to the task, which you may not be able to do if you had like one before the other. And if you're studying resting state or natural variations in brain states or certain spontaneous events like epileptic spikes, then that's something where you need to have the signals at the same time if you're going to want to directly correlate them. But simultaneous recordings also have some challenges and drawbacks, and one is that the data quality is compromised. It's actually mainly the EEG signal that suffers, so MRI is a pretty hostile environment for trying to record tiny scalp potentials. It also takes longer to set things up in the scanner, as well as before the scan starts, so you still have to prepare the cap, the EEG cap on the subject, and ensure that you have low impedances, a good connection between every electrode and the scalp. And that can increase your setup time quite a bit. You also need MRI compatible equipment that's gone through all the rigorous safety testing, and cleaning the EEG data after you've gotten it also takes some effort. So there are some trade-offs, and it really depends on your specific scientific question as to whether it makes sense to pursue these simultaneous recordings. So now let's discuss some things about the data acquisition and artifacts. So in terms of the EEG hardware, you need a system that's designed to operate safely and produce high quality signals in the magnet. So that means it shouldn't contain ferromagnetic material, and additional precautions are often taken in how the cap and the wires are designed in order to avoid heating of the tissue when RF pulses are applied during the MRI scanning. And on the MRI side, you also need to make sure that you're using pulse sequences that have been tested to not cause heating when there's an EEG cap. So this is a sort of generic block diagram of some of the components involved in the setup. So here's a person inside the scanner. That's their head supposedly wearing an EEG cap, and also a picture, you can see what it looks like. And this cap is connected through cables to the EEG amplifier, so that's this thing here. And the amps are typically placed either in the bore behind the head or right outside the bore, depending on what kind of system you have. And it's good to have short cables that connect these, and you also don't want the cables to vibrate in the acquisition, so you're going to want to try to stabilize them. The EEG amplifier is connected ultimately to a computer out in the control room, and this is usually done through a long fiber optic cable. And another thing that we're going to discuss in a couple of slides is that it's important to also synchronize the EEG acquisition with the MRI master clock. And this is because some of the artifacts that are produced during fMRI scanning are really high frequency, and you want to be able to sample those with no jitter so that you can try to remove them well. Another thing that's generally useful is to write certain markers to your EEG data, like the time at which every fMRI slice or volume was being acquired. That's also very helpful for removing artifacts in post-processing, as well as for being able to align these two data streams when you go to analyze them. And if you're presenting stimuli, you might also want to write in some stimulus markers to your EEG, and that's also helpful for analyzing things, making the timing precise. And so you can see this involves a number of extra components beyond just doing fMRI or EEG alone. And this is a rather simplified diagram because there are additional adapters and other things that connect these together. Okay, so next let's take a look at some signals. So here is some EEG data that's recorded while the person is inside the scanner, but before you've started to run the fMRI scan. And here's what happens when you start running your fMRI sequence. So unfortunately, you can no longer see anything that looks like good EEG. You just have this big wall of a high amplitude artifact. So what's going on? Well, this is due to what we're doing with the MRI scanner when we're making our images, which is that we're switching the magnetic field gradients very rapidly. And these time-varying fields produce voltages in the EEG amplifier inputs by Faraday's law. And this is a very high amplitude artifact, so it can be hundreds or so times larger than the neurally generated EEG signals. But something convenient about this is that the shape of this artifact is very repeatable from volume to volume or from slice to slice, because we're running the MRI gradients the exact same way each time. And in fact, if you were to zoom in on a time frame of one slice here, you can actually see this EPI gradient switching pattern very clearly in the EEG. So if we know then where all of our slices are happening in the data, then you can imagine building up a template of this artifact shape by averaging over multiple instances and then simply subtracting that out each time it occurs. So in fact, that concept, or some variant of this concept, which is called average artifacts subtraction, this is one of the main ways that people have gone about trying to reduce this artifact. In practice, the artifact isn't perfectly stable over time, so it drifts a bit. So for example, if somebody moves their head a little bit, it's going to change the shape because the pattern is spatially dependent. So in practice, this is typically carried out on a moving windowed basis. So that can be helpful for reducing the artifacts quite a bit, but in order for it to work best, it's important that your EEG acquisition is synchronized with the MRI clock. And this is because the EEG acquisition is undersampling the faster MRI gradient artifact, which is this gray waveform here. So if from one slice to the next, if you have even slight amounts of timing difference or jitter in where those samples are placed, then this could look very different from the viewpoint of the EEG. So imagine we're trying to build a template and we're capturing the green samples in one slice, and we get slightly shifted red samples in another slice, then we wouldn't get a clean template if we tried to average those. So as a result, our corrected EEG data would have more residual gradient artifacts, which is this in blue, compared to if these things were synchronized. And an additional note is that it's also useful to try and adjust the timing of your fMRI slice or volume TR so that it contains an integer number of EEG sampling points, which can also help to reduce artifacts. So gradient artifacts are the biggest type of artifact you'll find, and template-based methods work pretty well. So this section of data here has been cleaned of gradient artifacts. But now you can start to notice something else, which are these prominent deflections that seem to happen every time the person has a heartbeat, which is, you can see in the ECG signal also recorded here. So this is called ballistic cardiogram, or BCG artifact, and it's thought to arise mainly from slight pulsation-induced motion of the head or the scalp vessels. And it's much more pronounced if you're collecting EEG inside the strong magnetic field compared to outside the bore. As you can see, their amplitude is smaller than what we get with gradient artifacts, so they don't seem to corrupt the EEG as much, but they actually turn out to be pretty tricky to remove, and that's because they're more variable in their shape and from instance to instance. And so it usually doesn't work quite as well to use a template-based average to remove these. People usually do a little bit more signal processing. And for BCG removal, it's important that you do have a clean ECG signal available as well. And that's often just another drop-down lead from your EEG cap. So one of the main approaches that's been proposed for trying to reduce BCG artifacts is known as the optimal basis set technique. And the idea here is to use PCA, or principal component analysis, to construct a basis set or collection of shapes that well characterize the variation that you get in the shape of the artifact across a given channel. So to construct this basis, you could take a window around each occurrence of the artifact, as using the ECG as a reference, and input that to PCA, and then take the top K components to be your basis set. And then to clean things up, then for each instance, you could go and subtract out its projection onto that basis set. So here's from the study is data before applying the optimal basis set procedure, and then here afterwards, you can see that those deflections are much reduced, and you can clearly see the alpha oscillations. So this is one example of a BCG reduction technique. There have been other techniques and variations developed as well. Oh, and in addition to performing corrections only in software, there are also hardware-based corrections or modifications that can be done that help mitigate the artifacts too. So in this study, the idea was to put reference electrodes in the cap. So these are dummy electrodes that are physically attached to the cap, and they pick up BCG artifacts, but they're isolated from the brain's electrical activity through an insulating layer, which in this case is a shower cap, whereas the real EEG electrodes are given a path to contact the scalp. So these dummy electrodes generate helpful reference signals for subtracting out the BCG artifact from the EEG electrode signals. And here you can see it's quite effective and able to also reveal clear off oscillations when the subject closes their eyes. Another idea that's been proposed is to include these carbon wire loops on the EEG cap. And this is also a technique that can pick up non-neural effects that can be used as reference for obtaining cleaner EEG. So these can pick up things like the BCG artifact, as well as higher frequency vibration artifacts that you can get from the helium pump that's cooling the magnet. So this also adds noise to the EEG. So if we have these sensed and recorded, then you can also regress those out of the EEG data, such as by using a moving windowed regression, as was done here, to get a corrected EEG trace. So we know that MRI can introduce some pretty horrible artifacts on EEG, but one question you may have is, what about the other way around? So does EEG also produce artifacts on MRI? And yes, it can, though these effects tend to be less severe than the other way around. So one potential thing that may happen, and especially if you have a dense array EEG cap, like 256 channels, is that the bunch of cables that comes together as it's coming out of the cap, this can result in MRI signal dropout due to electromagnetic shielding effects, since it's between your RF coil and the brain. Another thing that can happen is that if the electrodes or the conductive gel have slightly different magnetic susceptibility from tissue, this causes inhomogeneity of the magnetic field and may also cause some signal loss here. So you can see these little bites taken out. It gets around the location of the electrodes. But it doesn't seem to go in very deeply beyond the skull. So I think it tends to be a small effect, at least at 3T. So that's, but one question is, what is the effect on bold contrast? Interestingly, it seems that even though you can get lower signal intensity near where the cables are coming out, the temporal SNR in bold fMRI seems to be much less impacted. And in this study, they found that this was due to the fact that there seemed to be less noise as well as less signal in those areas that were shielded. So if you calculate the functional signal to noise ratio, that loss is not as high as the loss in signal that you get on the numerator. So the upshot is that there is relatively small loss in bold contrast, which is good news. So assuming that we have cleaned the EEG data well, let's look at some examples of different studies and analyses one might do. And I'll only get to show a few examples here, but in the other talks in this course, you'll get to see many other examples. So I definitely encourage you to check those out as well. So as one example, EEG has a long history of being able to monitor certain brain states, such as different stages of sleep, where these different stages can be identified based on their fast electrical characteristics. So if you can get someone to sleep in the scanner with the cap on, which actually is easier than you might think, then we can look for windows of time where someone is likely in any particular sleep stage, like deep sleep or REM sleep, and then go to the fMRI data with its higher spatial resolution and look deeply into the brain at what's going on in certain regions or networks during these different conscious states. And this study looked at the default mode network specifically, finding that it seemed to be a reduction in the connectivity with anterior regions during deeper sleep. You can also look more extensively at region-to-region correlations in the fMRI data, both within and between different functional networks that may correspond to these different sleep stages. So in this set of studies, EEG was again used to identify when the person was likely in different sleep stages, and then you can go and take the corresponding sections of fMRI data to see what's going on in more detail. And here they did something else too, which is to use the simultaneous EEG fMRI data to train a classifier on the fMRI and see if you can predict EEG-based sleep stages using the fMRI data. So over here is fMRI-based sleep staging, which they calculated, they obtained basically by training a classifier on sliding window patterns of correlation in the fMRI data. So if you've trained such a model, then you may be able to then tell if someone is falling asleep in your fMRI scan, even if you don't necessarily have EEG collected at the same time. Another application is in studying spontaneous neural activity. So EEG oscillations in certain frequency bands like alpha, if you look at the amplitude of these oscillations, these don't stay constant over time, but exhibit these spontaneous variations. And these variations are thought to have functional significance in terms of behavioral and cognitive states. So fMRI isn't able to detect frequencies as high as alpha directly, but the slower variations in these envelope happen on largely similar timescales as resting state bold fluctuations if we look at their power spectra. So another type of analysis that's often done is to take the correlation between fMRI signals and the envelope of this EEG oscillation power in order to see which brain regions might be correlating. So here's an example, in fact, was one of the earliest human EEG fMRI studies. And here they took the EEG alpha power time course, the envelope, and they convolved it with HRF and used that as a regressor to ask on a voxel-wise basis which regions of the brain are positively or negatively correlated with that waxing and waning of the alpha power. And so they end up finding positive correlations in regions like thalamus and insula, and negative correlation in mainly cortical areas like occipital cortex. And again, actually, just like we might combine EEG and fMRI to see if fMRI alone can be used to detect sleep stages, we might also ask if fMRI can be used to detect more continuous fluctuations in brain states, such as alertness or arousal, that are indexed by varying levels of EEG power. And so this is one thing that we looked at recently. So what we ended up finding is that the expression of certain patterns like this in the fMRI data, that this may be used to detect these continuous fluctuations in EEG power that are linked with alertness. And we also find that the fMRI-based alertness measure could track a trial-to-trial behavioral variations, such as a person's reaction time during a scan. So I think it's promising that combining EEG and fMRI can help to reveal more information in fMRI itself that could be used to track different brain states. There are also other EEG features beyond the power in certain frequency bands that you can use to correlate with fMRI as well. So in this study, they derived a time-varying measure of phase synchrony, and they used that as a regressor to look at voxel-wise whole brain correlations. And they found that the phase synchrony of the upper alpha band was specifically linked with an array of frontal and parietal regions in the fMRI data, which formed a network whose function is linked with adaptive control. So the past few examples have looked at spontaneous brain activity, but there are also many interesting uses for EEG fMRI in tasks as well. And this diagram captures one example framework for analyzing the data during tasks. So after you remove EEG artifacts, then you might do some additional processing of your EEG, like ICA, to look at different features of interest, or perhaps not, depending on what measure you're interested in studying. And then for each individual trial in your task, you could extract some measure of the EEG that's varying across those trials, like the amplitude or the latency of an evoked potential, and use that to derive a regressor to analyze your fMRI data and find out which regions are tracking these kinds of electrical variations. So using this kind of approach, this was an earlier study that looked at an auditory oddball task, and they were focusing on trial-to-trial variation in the P300 ERP component. So here's the evoked response across different trials. These are sorted according to reaction time, and they were gathered inside the MRI scanner during the fMRI scanning. And so then you can try to correlate these with fMRI to try to figure out which regions are showing hemodynamic responses that are tracking these variations. And so I think this also illustrates how EEG can add really rich information to task fMRI, since it's giving us these electrical variations that we'd otherwise not have access to. And then there are also a number of clinical applications over the years that have driven the development of EEG fMRI. And one is epilepsy, where one important goal is in pre-surgical planning. So if fMRI can help us to localize non-invasively where epileptic activity is coming from, then it may inform which brain regions need to be surgically removed without first needing to implant electrodes into a person's brain. So by combining EEG and fMRI, you can try to identify electrical events of interest in the EEG, and then knowing where they are, you can map them using fMRI. So I wanted to wrap up there. I hope this helps provide some foundation for EEG fMRI. And as I mentioned, a number of other applications and techniques will be presented in the other lectures in this course. And here are just a few references that I could recommend for learning more about this field. I realize that the references are not complete here, so if you actually have trouble finding any of these, then feel free to shoot me a note. So thanks very much.
Hi. So it's great to be a part of this educational session focused on bridging electrophysiology and neuroimaging. I'm going to be focusing on how we can integrate signals from EEG and fMRI, particularly in the scenario where we're taking advantage of new developments in imaging to allow us to measure these dynamics at high spatiotemporal resolution. So what I'm planning to talk about, there's a brief outline here. First, I'm just going to kind of give a broad overview of the advantages and disadvantages of EEG fMRI and what types of scientific questions it's best suited for. And then I'm going to really focus most of the talk on discussing new higher spatial and higher temporal resolution techniques for imaging, how we can incorporate those with EEG fMRI, and how we might want to adjust the analysis techniques that we're using to jointly analyze these signals as we move to higher spatiotemporal resolution. So the beginning will be sort of maybe a broader overview of people perhaps interested in what kinds of science that they can or cannot use these questions for. And then the bulk of it is really going to discuss some of the techniques that are available and ways in which to best apply them for neuroscience questions. So I'm sure many of you have seen many versions of this figure before. Why might we want to do high spatial and temporal resolution fMRI? There's these plots that show that EEG is classically considered to have very nice temporal resolution, whereas fMRI has much better spatial resolution. And so the hope, of course, by integrating them is that you would manage to get essentially the best of both worlds, getting the high spatial and high temporal resolution. Of course, it's not quite as simple as that. It's not always straightforward how to integrate these techniques and how you could actually analyze the data to really bring you to that sweet spot where you have the high spatiotemporal resolution because of the use of both modalities. But ultimately, that's where we would like to move with this technique. So we know that the resolution of EEG is really excellent. It's used to look at event-related potentials locked to trials unfolding over, tens or hundreds of milliseconds. And it's also enabled us to look at oscillatory dynamics. So across different frequency ranges, we can look at either oscillations or spectral content across different frequency bands, spanning from very slow frequencies in EEG world, for example, around 1 Hertz, all the way up to gamma rhythms above 30 Hertz or even higher. So this gives us a nice readout of this kind of fast timescale temporal synchrony of neural activity. fMRI, by contrast, gives really excellent spatial resolution. So this is a plot from 10 years ago now showing the shift to really high spatial resolution fMRI, where you can start to not just image activity in specific brain regions, but you can actually image fMRI dynamics across specific layers of the cortex. And what's being shown here, for example, is how the spatial precision of the responses we can measure with fMRI can vary as you look from deep to superficial layers of the cortex. As we move from deep to superficial layers, there's both a neuronal difference and also a vascular difference that we're picking up with fMRI. So fMRI has really has had a lot of kind of use as a really high spatial resolution technique. Classically, it's considered to have pretty poor temporal resolution. So this is an example of a typical hemodynamic response. You can see even if there's a very brief event of neural activity shown in this rectangle here, you have a very slow and slowly unfolding evolution of the hemodynamic response, which is ultimately what we're measuring with fMRI, peaking, maybe 5 seconds later and really spread out in time. And so because of this hemodynamic aspect, which is what we ultimately measure with fMRI, it's often been viewed to have quite poor temporal resolution. Maybe hundreds of milliseconds and not the tens or individual milliseconds that's achievable with EEG. So in addition to having different resolution, EEG and fMRI are really measuring kind of fundamentally different things about neural activity. So EEG is essentially measuring synchronous activity of spatially structured neurons that are undergoing coordinated activity together. fMRI is measuring a downstream effect of neural activity, which is that when neurons are active, it triggers a cascade of events that ultimately results in increased blood flow to an area, and so increased blood oxygenation. And so because of this, there may be many kinds of neural activity that are actually invisible to one of the modalities and only visible to the other one. So you can imagine, for example, say you have an oscillation in neural activity. Maybe these neurons are all firing together, which you can detect in the EEG. It could be that when you see that EEG rhythm, say an EEG alpha rhythm, maybe the actual mean firing rate goes down and the neurons happen to oscillate synchronously, or maybe the mean firing rate went up. And so you can imagine that the same signature on the EEG could give you either a positive or negative change on fMRI. And so this kind of points to a really useful aspect of combining the two of these, right? There's many different aspects to neural activity that we can infer if we actually make two different kinds of measures, both about the synchrony and about the overall magnitude of neural activity change. A second aspect of neural activity that these really sample differently is that EEG is very focused on surface activity. So we place sensors on the scalp, and they're sensitive to neural activity that's relatively close by. Whereas fMRI has this whole brain resolution that allows us to measure, for example, tiny deep brain structures like this visual thalamus nucleus that I've highlighted here in red. And so this reflects that in addition to their different spatial temporal resolution, they really are giving us complementary information about the nature of neural activity and the spatial properties of neural activity that's very useful to combine. An added benefit to doing simultaneous EEG fMRI is that it can give you a lot of consistency across your experiments. So you might think, well, why not just do my study with EEG and do my study with fMRI? And in many cases, that's probably the right answer. But there's many scenarios where it's not. So for example, task conditions are really different in a loud, noisy scanner than they are in a quiet EEG room. Somebody who does a task first in one setting and then again on a second setting may have learned, have training effects or novelty effects that vary across those two sessions. And also brain state is very dynamic. And so there's no guarantee that when somebody did the task in one context that they were using the same cognitive strategy or that they were paying as much attention as they would the other time. So across all of these different factors, you can often better pinpoint neural activity if you do both of these simultaneously. EEG fMRI is also really excellent for looking at single trial analyses. So anything where from trial to trial, something about the phenomenon that you're measuring is varying, you really need these measures to be simultaneous in order to link what's happening. So for example, if you're looking at vigilance changing over time, maybe something like visual illusions or other bistable perceptual tasks, if you care about attention and how that's modulated on a trial to trial basis, or if you actually want to directly bridge neural activity across EEG and fMRI signals and understand, for example, why trials with larger bold responses may also have different aspects of EEG activity. So across all of these, you can see that EEG fMRI and integrating these two types of information may give us a lot of useful information about brain dynamics, particularly dynamics that are really unfolding over time and aren't necessarily constant over an entire experiment. Another use case that's very kind of applicable for this is if you want to look at changes in brain state. So for example, across sleep, we can see that people, this is showing the EEG of people in different sleep stages, and you can see that there's really different brain states that emerge. And this is EEG in the case of epilepsy, where the stars are highlighting interictal events. So by doing these things simultaneously, you could imagine that you could actually use fMRI to look at brain-wide activity patterns, for example, locked to individual events identified in the EEG. So EEG fMRI presents a benefit also for spontaneous neural dynamics. So I listed all the great reasons to do EEG fMRI. Why would we not do it? There's many great reasons not to as well. And so I think it's useful to consider for a given question whether or not this technique presents more problems than it does advantages. So very pragmatically, the setup time for an EEG fMRI experiment is far higher than for a single modality alone, and it's very easy for one component of that setup to fail. So there's an added burden that comes with that. EEG quality is significantly affected by being recorded in the scanner. And although I'm going to cover lots of techniques for improving that issue, the reality is that the EEG will never be of the quality that it would have been if it was not recorded inside the scanner. Experimental designs are often not well suited to EEG and fMRI. So for example, the way we time our task presentations for one modality versus the other is really different. EEG, you often have lots of trials coming rapidly together, while in fMRI, you might space these out to allow more time for hemodynamic responses to recover. So when you do them at the same time, you're really constraining that you can only use a given task, and it may not actually be the right task for one of those two modalities. And so because of this, many studies, it might be really useful to combine the information from these two, but it might be just as useful to do that with separate EEG and fMRI experiments. And so it's really worth considering whether the question you're interested in has this spontaneous dynamic element or single trial question, or something about the kind of context and task environment that you really want to hold constant as you're doing your analysis. Finally, I'll touch on this more in detail in the end, but even once you do acquire these simultaneous data, how to actually take advantage of that simultaneity in your analysis is not always straightforward, and so that requires some thinking as well. Okay, so moving now into this concept of high spatiotemporal resolution. So if we wanted to not just do EEG fMRI, but do EEG fMRI while taking advantage of really the sort of edge of techniques of what's possible for fMRI, what might be available to us? So ultra high field fMRI, by that I mean fMRI with magnetic fields that are stronger than the often used 3 Tesla magnetic fields. So this is scanners at 7 Tesla and even above. In past years, there's been an increasing number of, even 9 or 10 Tesla scanners coming online. So as the magnetic field becomes stronger, the signal to noise ratio of our images becomes better. You can get quite a large effect from this. So typically, in a 7T scan that I do, I see twice as much SNR as I would get in a 3T scan. And so that boost in SNR alone is extremely useful for detecting subtle aspects of brain dynamics. But in particular, this is often used to increase the spatial resolution of our imaging. And I think these images give a really nice illustration of that effect. So this is showing here very high spatial resolution data acquired at ultra high field. And then you can see as this data is spatially smoothed, how this changes the kinds of effects that you would measure. And so you can see that if you're doing low resolution imaging or applying a lot of spatial smoothing, you can get very strong activation patterns, but they're really very blurred relative to, for example, the cortical gray matter or the actual kind of sources of origin of that signal. If you're using ultra high field imaging and high spatial resolution, you can actually see this really intricate structure where the activity can often really be carefully confined to sort of that cortical surface. And you can also see that there's very distinct patterns across the layers or across different cortical regions that become obscured. But tellingly, right, the statistics are much smaller in this original one than they are in the blurred one. So it really requires like an increased SNR to be able to push this high spatial resolution imaging. So once you are doing ultra high field fMRI or doing high spatial resolution imaging, what comes into view? So the first is, as I mentioned before, there's been a really intriguing shift to using a very high spatial resolution fMRI to look not just activity in a cortical region, but across layers. So this is an example of a very recent nice study that was done using both bold fMRI, but also vaso fMRI, which is a more CBUV sensitive technique, and using different manipulations of touch and motor activity to try and selectively activate either deep or superficial cortical layers because of the different computational roles of each of these layers. And they were able to detect distinct dynamics in deep and superficial cortex using this very high spatial resolution fMRI. The other thing that really comes into reach with us with high resolution imaging is the deep nuclei of the brain. So a lot of fMRI has focused on cortex, but there's also been several studies showing imaging of really fascinating and key brain structures like the thalamus and the brainstem. These structures are composed of dozens or even hundreds of really tiny subnuclei that have very different functional roles. And so this is an example just from the visual system of using high resolution imaging to localize the lateral geniculate nucleus of the thalamus, which is a primary visual nucleus, and even this very tiny structure, the superior colliculus, which is the visual part of the brainstem. And so you can see these very small regions imaged here with 1.1 millimeter voxels, but even lower are sometimes possible with high spatial resolution fMRI that are coming into view as we increase the SNR of our imaging. So these are really interesting to do in concert with EEG dynamics. So there's a rich literature on the thalamus and the structure of different cortical layers, and the fact that the interconnection of thalamus and distinct cortical layers is really critical for synchronizing activity across cortex and modulating both low and high level cognitive processes. So everything from kind of sensory perception to working memory and attention is reflected by the coordination of different cortical areas and its interactions with distinct nuclei of the thalamus. So the EEG rhythms that we measure at the surface are really linked to the computations that are taking place across these large-scale distributed circuits that involve these mesoscale structures, the cortical layers and the thalamic nuclei. And so by integrating high spatial resolution fMRI with EEG, it really brings this picture into focus more, where you're able to actually now begin to tackle how activity measured with fMRI across these tiny structures may relate to EEG dynamics that you can measure at the same time. So I think that this interaction is a very intriguing application area for high resolution EEG fMRI. So that's high spatial resolution fMRI. But what about high temporal resolution fMRI? So over the past decade or so now, there's been a real shift in the speed of fMRI. It used to be that to take a whole brain image, it would take maybe 2 to 3 seconds. You would have to kind of step through the brain slice by slice, and that whole process of assembling an image would take 2 or 3 seconds. There's now been the development of simultaneous multi-slice methods, which allow you to take multiple slices at the same time. So if you scan, for example, 8 slices at the same time, you can actually acquire image 8 times faster. And so because of that, it's now possible to do fMRI, the whole brain fMRI, in just, for example, 400 milliseconds or so. So we can scan really quickly now. And there's a couple of good reviews on these techniques that are listed at the bottom of the slide. But the question is, well, how fast are the signals we're actually measuring? We know that the canonical hemodynamic response function that's often used in fMRI analyses is slow. It peaks maybe 5 seconds after neural activity. It unfolds slowly and blurs away a lot of these fast signals. However, there's been many different resting state fMRI studies now that have shown that, contrary to the predictions of this canonical model, bold signals do not exist just below 0.1 or 0.2 hertz, where we might have predicted them to be. There actually seems to be interesting information content in the resting state bold signal well above that 0.2 hertz range. And here's a series of papers that you can go to for more information on this topic. In addition to these resting state studies, in our work, we've used task studies to provide neural activity at known given oscillation frequencies and test how fast are we able to go in recovering those oscillations in the fMRI response. So while conventional models predict that you shouldn't really be able to see signals above 0.2 hertz, you can see here that when we averaged the visual response to a visual stimulus oscillating at 0.2 hertz, we saw a very clear response in the fMRI signal. And we could track this all the way up to at least 0.75 hertz. These signals were an order of magnitude larger than predicted by canonical responses, really suggesting that there's surprisingly fast dynamics in the fMRI signal that you can capture when you start to image at faster rates. In addition to just being able to detect ongoing dynamics that are at higher temporal resolution, scanning quickly can also enhance detection of, for example, the onset times of neural activity. So this is a really interesting study demonstrating sensitivity to differences as small as 100 milliseconds in when activity began in different cortical regions. In addition, if you had a very kind of brief neural event, you can imagine that scanning very rapidly could help you resolve the timing of responses to that brief event. So to briefly summarize this first part on the high spatial temporal resolution fMRI, scanning quickly and with high spatial resolution really kind of enhances what we can do when we're bridging imaging data with EEG. Firstly, it has the potential to bring what we're measuring into the same spectral band. So typically fMRI and EEG were in completely different temporal ranges. But now in this sort of 0.1 to 1 hertz band, we actually have a dynamic where we're able to measure both with fMRI and with EEG. And this is a really interesting temporal band, right? So 0.1 to 1 hertz is sort of hundreds of milliseconds. It's a temporal range over which many interesting cognitive processes unfold, anything from, for example, language processing to, high-level attentional control. And so these are the temporal ranges where, when we do high spatial temporal EEG fMRI, we now have access with both modalities. It also enables us to begin to link oscillatory dynamics and EEG events to relatively fast timescale brain-wide activity measured with fMRI. And we can also begin to uncover activity both in cortical layers and in small individual deep brain nuclei and understand how those are linked to electrophysiological activity as well. So this, I think, is the potential that's offered by high spatial temporal EEG fMRI. What are the challenges that we might encounter, and how might one need to change experimental paradigms and analysis streams to try and take advantage of this interesting technique? So the first kind of key consideration to talk about is just safety, participant safety in the scanner. EEG quality is really modulated by these choices, and so I'll talk briefly about some strategies that people can use to improve EEG quality when using these techniques. And then finally, some analysis streams that can really take advantage of these to try and uncover new aspects of brain activity. So the key safety consideration for EEG fMRI is the fact that whenever you introduce an object with metal in it inside a scanner, there comes with that a risk of heating. So EEG fMRI is and should always be done with EEG caps that are specially designed to be used in the scanner. So they don't have ferromagnetic components. They're not attracted to the magnetic field. But it's usually not possible to remove all metal. And so with that comes a concern about heating of the electrodes. And so any given EEG cap for fMRI will have guidelines that come with it for what kinds of scans you can do to safely conduct the scan and avoid any risk of heating. It's important to note that when you increase the spatial and temporal resolution of fMRI, with that can come increases in SAR and power deposition that are really important to take into account when you consider what effect that will have on heating. These plots are, for example, showing temperature increase over time during scanning, either without an EEG net or with an EEG net. In this case, showing that there's, in fact, no clear effect of the EEG net for this particular scan, and that this is going to be safe to use on a participant. In general, when you scan even without EEG, there is a temperature change in the bore. So, taking that into account is useful. If your whole room is getting warm, that's not a sort of EEG specific effect. But manufacturers have guidelines for this, and this is also something that can be measured directly to understand the heating effects of high spatial temporal resolution on EEG. A second really significant challenge for doing, for example, 7T imaging with simultaneous EEG is the artifacts. So a typical EEG cap, firstly, at 3T will induce some signal dropout, but in general, it's not really a major problem at 3T. As our magnetic field increases, this becomes a much more serious problem. And so you can find that there's regions of the images when using kind of conventional EEG caps that may be much more challenging to image with this EEG cap kind of shielding signal from that brain region. I'd say the biggest problem, though, is really the quality of the EEG. So I'm plotting here some data I acquired, EEG at 3T and at 7T. And these spikes that you're seeing here that are rhythmic, this is the ballistic cardiogram artifact. This is an artifact that's locked to each beat of the heart with EEG. And you can see here, these are on the same scale, that this artifact is many times larger in the 7T EEG. And in this case is really obscuring anything that we might hope to see in our ongoing EEG signal. So removing these cardiac locked and motion artifacts in the EEG becomes a very important problem at 7T, even more so than it was at 3T. Some strategies people are now developing to try and address these approaches. Firstly, there's very exciting kind of technological work being done on how to design new caps for EEG at 7T. So the next talk by Dr. Giorgio Bonmassar is going to address some of these new technologies for EEG fMRI at 7T. There's also signal processing strategies. So I think that Dr. Catie Chang in her previous talk discussed, for example, the use of basis sets to model what the heartbeat artifact looks like and take that out. That kind of approach often works pretty well at 3T. It may not be sufficient at 7T, where the noise is so much larger that in addition to this kind of signal processing strategy, you need to bring other techniques into play to try and clean your signals. So an example of this is this harmonic regression technique. This is showing a spectrogram of clean EEG and then EEG, for example, at 3T. And you can see that the artifact has this very clear striped structure in the spectrogram. So this signal processing approach essentially tries to recognize those stripes and use that information to recognize the artifact and remove it from your signal. There's also a couple of great techniques that actually directly measure the noise and then take that into account to try and model the noise and subtract it from the signal. So this shows a reference layer, modifying your EEG cap such that some electrodes, instead of recording from the scalp directly and recording both noise and artifact, some electrodes are isolated from the head and just record the artifact and not the signal. And because of that, you have a very nice model you can build of what the artifact looks like that you can take into account to do your signal cleaning. There's been a lot of great work, particularly for EEG fMRI at 7 Tesla, done by Joao Jorge and his colleagues. They've focused, for example, on developing things like carbon wire loops, where they essentially add on extra wires to their EEG caps and use that information from those isolated electrodes to really, again, measure the noise, and then you can use that noise and remove it directly from your signal. And this kind of approach is really ideal once you're moving to 7T EEG fMRI. I also just want to mention a couple of really important things to be extremely careful about when either conducting or just reading and interpreting an EEG fMRI study. The first is that it's very important to keep in mind that the strongest component of both signals is going to be motion artifact, and that because this is simultaneous, the motion is going to take place at the same time in both data streams. So if you're just correlating, for example, EEG power in a specific band and bold activity, when somebody moves, you're going to have a spike in EEG power, and you're also going to have a rapid change in your bold activity. And so any analysis that doesn't take into account this motion correlated noise is going to be very vulnerable to motion related artifact. And there's a nice paper discussing this by Fellner et al. This is a neuroimage that is great reading for kind of what to look out for in this approach. One thing to keep in mind is that these motion correlated artifacts can look very physiological. And so it's important to really try to exclude motion in your analysis stream to avoid being led down the wrong road by these artifacts. The other really important thing to keep in mind is that already with fMRI, there's a lot of choices that can be made in an analysis stream. To do successful high spatiotemporal resolution EEG fMRI, it takes a lot of work on the data cleaning side, and it's really important to make sure that you don't end up with a circular analysis such that you end up tweaking your pre-processing to get a certain kind of result. There's so many degrees of freedom in how to clean the data that it's important to keep in mind strategies such as developing your cleaning pipeline maybe just on one subject and then not touching your other subjects and then applying it to them, or using some kind of strategy where you make sure that the choices you're making about how to clean one data stream aren't ultimately ending up with some kind of circular analysis where you're kind of pre-concluding the results that you'll see at the end of the day. Once you've kind of taken into account the data cleaning, there's other kind of considerations to keep in mind. So when you have high spatiotemporal resolution fMRI, you'll have different noise properties. For example, you may alter the relative ratio of thermal to physiological noise. As I described before, we have lots of altered noise in EEG. And then a final point that I think is really important to keep in mind also is that there's really going to start to be a need for updated models of the hemodynamic response when scanning at high spatiotemporal resolution. So what do I mean by that? We know from a large existing literature now that the hemodynamic response function is not the same across the entire brain. In particular, we know that it's faster in the subcortex, for example, in many structures of the thalamus and the brainstem. It's faster in the deeper layers of the cortex than it is in the superficial layers of the cortex. And it's also faster when you have brief neural activity instead of long neural activity. A stimulus that lasts just 100 milliseconds will give you a faster response than a stimulus that lasts longer. So here's just some visual examples of this. These are, for example, hemodynamic responses in visual cortex in response to a long stimulus here in red or a short stimulus in blue. And you can see that the time to peak really becomes faster and faster as your stimulus gets faster and faster. But if we also look across visual cortex, visual thalamus, and visual brainstem, you can see that this response is also getting faster and faster in the deep brain structures. There's also a ton of work showing that what I show here Sierra et al., for example, showed that the deep gray matter has a much faster hemodynamic response than the surface gray matter. And in particular, the surface tends to have really large vessels, for example, large draining veins, that have the slowest and largest responses. There's also lots of great work on the nonlinearities of these responses to different stimulus types. So once we start imaging at high temporal resolution or at high spatial resolution, these effects that were always there and were always present in the data, but were maybe kind of harder to see with low resolution data, started to really come out very strikingly. And so it's pretty critical, I think, to keep this in mind to really develop analyses that apply across the brain. So a really common approach, for example, to analyzing EEG fMRI data is to use the EEG to either maybe identify specific events or to extract, for example, power in a specific band, say maybe the amplitude of alpha power over time, and then to convolve that event or to convolve that power with a hemodynamic response. When that analysis is done, that's really assuming a specific shape for this hemodynamic response. And so to really kind of take advantage of high spatial temporal resolution imaging, it would be good to use a model for the hemodynamic response that's flexible and that allows you to capture either faster or slower dynamics, since we know that depending where you're looking in the brain and depending how fast your neural activity was, the correct model for this is going to change. In addition to kind of pre-prescribing what kind of response you expect to see and what kind of coupling you expect to see, EEG fMRI also gives us the opportunity to use kind of more unsupervised or data-driven methods to try and discover, regardless of what we might predict, what coupling do we see and just appears to be present in our data. So there's lots of interesting work, for example, on using joint ICA across the EEG and fMRI data to discover which aspects of these signals are coupled and which aspects of these signals are not, and to really take advantage of the kind of joint properties of the EEG and fMRI to understand this both in time and in space. There's also interesting work, for example, using Markov models and PCA to identify underlying states that are present in the data without having to prescribe ahead of time what those might be. So for example, in transitions into sleep, discovering from the data alone, what distinct states seem to be present and which states tend to transition into other states. And then, of course, there's, with the explosion of deep learning, there's also beginning to be quite a bit of interest in deep learning for EEG fMRI. And there's pros and cons to trying to take this approach. So EEG fMRI is very complex, structured and high-dimensional data. And so in that sense, it's a really great candidate for some of these techniques. But in another sense, it's really not. And that's that these data sets are so hard to acquire that, in general, we don't have sort of human connectome-style projects for this kind of data yet. We don't have really large N data sets with lots and lots of data. We tend to have pretty small studies still. And so for deep learning, it's very data hungry. And so that's a challenge, I think, particularly for trying to kind of apply these techniques for EEG fMRI. But there's still strategies that one can use to try and kind of take some of the power of those techniques. There's two questions for EEG fMRI that they're probably going to be quite useful for. One is that we have very high dimensional data, and there's probably many interesting dynamics that are really existing on latent lower dimensional manifolds that can really be perhaps identified and better extracted by some of these data-driven techniques. And then the other thing is that we have very structured noise in these data. And that's another real opportunity for these kinds of technologies. And so an example of a recent paper focusing on this is trying to use kind of deep learning style approaches just to identify what the BCG noise looks like and remove that automatically from the data. And they seem to be seeing success with this approach as well. So I think that this is a really developing area. It's going to be interesting to see what comes out in the next few years. Just to very briefly touch on a couple example applications of high spatial temporal resolution EEG fMRI. It works best when your phenomenon of interest is spontaneous, variable across trials, or is actually about the coupling of the signals itself. And so an example is using EEG fMRI to look at laminar dynamics, like this paper by Shiringa et al., where they were looking at attentional dynamics on single trial levels, really across cortical layers, very ambitious measurements to be making. And in our own work, for example, we've used this to look at sleep. So using fast fMRI as people are transitioning into sleep to not just look at low frequency changes in fMRI, but because we image so quickly, we can actually take a spectrogram like one normally would have done for EEG and apply that analysis to the fMRI signal itself and see these really striking 0 to 1 hertz power increases in the fMRI signal in somebody who's just transitioning into light sleep, which really kind of opens up the temporal spectrum of activity that we can now look at with fMRI. So to conclude, EEG fMRI provides high spatiotemporal resolution and allows us to measure really multiple aspects of neural activity. The increasing speed and sensitivity of fMRI offers new opportunities for multimodal imaging. And I think the joint inference for EEG fMRI is a really interesting and active field that we're engaged in as well, and that is going to be interesting to continue to push the boundaries of how we can analyze these joint data to really take advantage of trying to image the brain at mesoscale, both spatially and temporally. And so I think there's many interesting problems in neuroscience and neural dynamics that are going to be really interesting to pursue using these approaches. So I hope that I'll have an opportunity to take questions online, and thank you all for your attention.